All right, good evening, folks. And today or tonight, I'm going to be doing a part 2 to Intro to Developmental Biology, the learning objectives, concerning differential gene expression in early embryos. So part 1 focused on, I kind of did a in reverse. I focused on major developmental events, which is actually part 2 of the learning objectives, but it was actually quicker, and I figured that if you only needed to see that one, it was nice and quick, around like 9 minutes, I think. So this one is going to be concerning differential gene expression in early embryos. So that's part 1 of the learning objectives. Okay. So compare and contrast proliferation and differentiation. So proliferation is going to be like the multiplying of cells, right? So pro is like 4, so if you proliferate something, it's like multiplying. So that the same is for cells. You create a great deal more of cells when you have cell proliferation. And cell differentiation is going to be when you have cells. Cells are going to be told kind of where their certain fates, what their certain fates are. So cell fate is determined. So they kind of go hand in hand in that in order for proliferation to happen, differentiation needs to occur first, right? And so I'll give you an example. So osteocytes are mature bone cells, and so you wouldn't want osteocytes in your liver. You wouldn't want bone cells in your liver because those cells are meant to be for bones, right? And so you wouldn't want mature bone cells in your liver. And so in order for a great deal of cells to be produced, you need differentiation to happen, to say, okay, these are osteocytes, or these are hepatic cells, or nerve cells, you know. So you need those things to happen in order for proliferation to occur. Okay. Understand the levels of organization of organisms starting with cells. Okay, so obviously every organism is different. However, there's some similarities. So if you have taken biology before, or maybe this is your first time, maybe you've heard of the field of comparative embryology. So what that is scientists will actually kind of take pictures of various types of embryos and compare them. And so comparative embryology was really big for evolution. And so, to prove that evolution was a thing. And so the point I'm getting at is that the levels of organization throughout organisms are very similar. If you look at pictures of human and shark embryos at a certain point, they look almost identical. And so across species, it's very similar. The levels of organization. So first you're going to start with a zygote, which is a fertilized egg. You're going to have a lot of cell division that occurs during the first 4 weeks of embryonic development. And at some point within those first 4 weeks, you're going to get a blastocyst, which is responsible for gastrulation. I talked about that in the other video, but and then after that, you're just going to have, after gastrulation, you're going to have more cell division, etc., until you finally end up with an organism. So the core at the levels of organization is cell division and appropriate cell division. Okay, so define a zygote. I just talked about this, but a zygote is a fertilized egg. So sperm and egg come together. That's a zygote. Describe the 3 major ways in which a zygote is transformed from a single cell to an adult organism. Well, 3 major ways. Well, at the core of this is cell signaling. So I'm not entirely sure what they're getting at here, but what I'm presuming is they mean cell signaling processes that really determine what will happen to that zygote. So without cell signaling processes and growth factors and etc., you'll have a very rudimentary cell. So cell signaling really should be at the heart of that. Describe what is meant by differential gene expression. Okay, so across a variety of organisms, you see genes, right? Similar genes. However, you may not have those genes expressed the same. So differential gene expression is due to cell signaling, which is going to signal for different genes to be expressed. Describe the two main processes that cause embryonic cells to head down different pathways. I'm presuming they mean, for this question, I'm presuming they mean cytoplasmic determinants and cell signaling. Yeah, those are the two main processes that I can think of. Describe what cytoplasmic determinants are, their origin, and how they contribute to differential gene expression when a zygote divides. Okay, so cytoplasmic determinants are maternal substances in the egg. So figure sperm is going to be released. That sperm will swim up. It will meet the mature follicle that will go down the fallopian tube. They'll meet in the fallopian tube, they'll go down, and they'll implant on the uterine wall. Okay, and so when that happens, right, you get a zygote, and so within that zygote, you're going to have an egg, right, and a sperm, and within that egg, you're going to have cytoplasmic determinants. So those are maternal substances, right, that are going to separate unevenly. So when you have that massive cell division that I just discussed in the first 4 weeks of embryos, a tremendous amount of cell division will happen, and those cytoplasmic determinants will separate unevenly, and depending on how they separate, that can influence gene expression. Describe the meaning of induction in embryonic development. Okay, so this is all part of a larger concept called cell signaling. So cell signaling pathways consist of kind of three main parts. You've maybe heard of this in AP biology, or maybe this is your first time hearing about it. But you're going to have a signal, transduction, and response. And so transduction is really when that cell signal is processed. The response is obviously the intended effect. And so induction is just the process of taking in of that cell signal to be interpreted for the appropriate response. Compare and contrast determination and differentiation in development. Okay, so determination is going to be when a cell is assigned a certain fate. Differentiation is the molecular processes that commit that cell to that certain fate. Describe a master regulatory gene. A master regulatory gene is going to be a gene that basically is going to commit cells to their certain fates. Define apoptosis. Okay, so apoptosis is going to be programmed cell death. Describe the characteristics of cells undergoing apoptosis. Well, they're dead and or non-functional cells. They need to not be there anymore to be replaced by new cells. And give examples of the importance of apoptosis in specific developmental events. Okay, well, so in the neural system, and for example, morphogenesis, which is kind of the formation of limbs. It's very important in embryonic development for normal morphogenesis and normal nervous system development. Describe the three major axes of the 3D body plan. Okay, so the three major axes, so you're going to have right and left, dorsal, ventral, and I believe it's posterior and anterior. Okay. Define pattern formation with respect to animals. So pattern formation is basically focused on, how the body is organized, and there are certain regulatory genes that impact pattern formation. Describe the basic body structure and axes of adult and larval fruit flies, or Drosophila. So the basic body structure you're going to have, like similar to all arthropods, which is basically a fancy word for insects and crustaceans. Within arthropods, you're going to have kind of this segmented structure, so a head, a thorax, and kind of this tail area. And then the axes, it's very similar. You're going to have, right and left, dorsal ventral and posterior anterior. Okay. Describe key events in Drosophila development from developing egg to the larva. I don't know this offhand. However, they have a really great example. Dr. Apasami's PowerPoint has a really fabulous example of this. Primarily what you need to know is that, there's a sequence of events, right, which kind of results in egg maturation. Then that egg is going to be fertilized, which results in a rudimentary Drosophila organism, which will result in an adult Drosophila organism. So that's basically the synopsis, but for more details you can see Dr. Apasami's PowerPoint. It's a really great in-depth explanation. Understand the organization of homeotic genes in fruit flies and how the linear pattern of homeotic gene expression is related to different segments of the fruit fly larva and the adult body form. So homeotic genes are really responsible for correct pattern formation, there's also really great pictures in Dr. Apasami's PowerPoint, but basically what you need to know is that weird things, that mutations in these homeotic genes, scientists have played around with this, will result in unusual body formation or pattern formation. Define maternal effect gene and egg polarity gene. So the maternal effect gene it's quite interesting. It's a gene that will affect organisms despite cell signaling. So it's a pretty determined gene. And then egg polarity gene that can also be found on Dr. Apasami's PowerPoint, but from my understanding, it's responsible for correct pattern formation as well. Okay, so describe the pattern of expression of a maternal effect gene product, mRNA and protein bicoid, in the mature, unfertilized fly egg and early embryo. So a maternal effect gene, in this case we're discussing the bicoid gene. So if it's in a bizarre place, you'll result in Drosophila with two kind of tails, I guess is the best way to describe it. So you'll have like this kind of weird round fruit fly that doesn't have a normal head and tail axis, more or less. Describe the structure and the mechanism of action of bicoid. So bicoid is the maternal effect gene that is going to result in kind of weird body patterning. Define morphogen gradients of morphogens. Okay, so a morphogen is going to be kind of any chemical substance that will affect an embryo, and gradients of morphogens are going to be like, if you think about it, we discussed in our lab, concentration gradient is really important. So depending on how much some of the substance there is, so figure a concentration gradient of morphogen. The more exposure to chemical substances that will affect the embryo, the higher likely mutation rate that you will have for these embryos. So I hope that was helpful. That is part 2, even though I'm discussing part 1 of the worksheet. So if you have any questions about what I've said in this video, please feel free to shoot me an email, and stay safe and stay healthy.